Good morning, students. Today we are going to start with zoology. Zoology first lesson, reproduction in organism. In 11th standard we have learned about the characteristic features of living organism, their classification and some of the main systems like digestive system, respiratory system, excretory system, endocrine system, nervous system, etc. Now, in this lesson, we are going to learn about the reproduction. So, before that, let us recall the characteristic features of living organism. So, what are the characteristic features of living organism? So, they are born, birth, they grow, growth, they develop into adult, they mature, and they are able to reproduce, and finally, they will die off. So, now define reproduction. What is reproduction? So reproduction is the fundamental feature of all living organisms, we have already seen. It is a biological process by which organisms can produce their young ones. So young ones are produced by the process called reproduction by the adult individual or matured individual. Now, what are the significance of reproduction? Why should the reproduction take place? So reproduction has to take place for the continuation of the species. So every species of organism has a definite lifespan. So after their lifespan they will die and they must be replaced by their successive generation, and this happens through the reproduction. So this world has seen many generations of different kinds of people. So next significance is that introduction of the variation in organism. So what are variation? So the individuals which are produced by the parent vary from their parent as well as among themselves. This is because of the meiotic division, crossing over and exchange of chromosomes taking place in the sexual reproduction. Then evolution of their own kind. In the same way, so slowly little variation leads to the different kind of species. New species are evolved because of the variation, and this is what we call as evolution. The best example would be modern man evolved from the early man. So these are the three significances of reproduction. The next one is the basic features of reproduction. So basic features of reproduction, all the living organism has to undergo certain changes in their body. So all the modes of reproduction have some basic features. These have to take place. Then only they can reproduce. What are they? So first one is the synthesis of RNA and proteins. The second one is the replication of DNA. So replication of the DNA means DNA makes a copy because of which new cells are produced. So this give rise to the cell division and growth of the organism. So and also this leads to the formation of the reproductive units and fertilization to form the new individual. So these are the steps or the basic features the individual has to undergo before the reproduction. So I repeat, the synthesis of RNA and protein molecules, replication of the DNA, cell division and growth, formation of the reproductive units and fertilization to form the new individuals. Now let's see the modes of reproduction. What are the two modes of reproduction? So all the reproduction can take place in these two modes only. First one is asexual reproduction and second one is the sexual reproduction. Now let us see first what is asexual reproduction. For the asexual reproduction, single parent is enough. So reproduction by the single parent without the involvement of the gamete. You know what are gametes? The male cell and the female cell. Male cell is called the sperm and the female cell is called the ovum. And without this involvement of the gametes, if the individual is able to produce the young one, we call this mode of reproduction as the asexual reproduction. Here the offspring produced will be genetically identical to each other. They will not vary from one another. Then asexual reproduction is usually happens by two types of cell division. They are amitotic and mitotic cell division of the somatic cells. So somatic cells means body cells or vegetative cells. Hence it is otherwise known as somatogenic reproduction or blastogenic reproduction. It is very common in the members of Protista So the primitive organisms, protista, bacteria and archaebacteria. The second type of more reproduction is called as sexual reproduction. When two parents participate, we need a male and a female parent for this. Two parents participate in the reproductive process involving two types of gametes. We have already said ovum and sperm. Then it is called as sexual reproduction. So I repeat the definition: when two parents participate in the reproductive process involving two types of gametes like ovum and sperm is called as sexual reproduction. So let us see the different types of asexual reproduction. The different modes of asexual reproduction seen in animals are fission, sporulation, budding, gemule formation, fragmentation and regeneration. Now let's see first fission. What is fission? Fission is the division of the parent body into two identical daughter individuals. The cell will divide into two so that each piece will become a individual. So identical daughter individuals are produced because of the fission. And this fission also can be classified into binary fission, multiple fission, sporulation and strobylation. So let's see first binary fission. What is binary fission? Binary fission, the parent organism divides into two halves and each halves form a daughter individual. Here first the nucleus divides amitotically or mitotically. So the division of nucleus is called as karyokinesis. Karyo means nucleus, kinesis means division. So karyokinesis, and the division of the cytoplasm takes place. This is karyokinesis is followed by cytokinesis or the division of the cytoplasm. Here the resulting offsprings are genetically identical to the parent. The parent also identical to the individuals and the individuals also will be identical to each other. So all will be similar. So depending on the plane of fission, the binary fission can be classified into simple irregular binary fission, transverse binary fission, longitudinal binary fission, oblique binary fission. Let's see the first one, that is simple irregular binary fission. Simple irregular binary fission takes place in amoeba. So the nucleus divides mitotically. The cell then constricts in the middle, so the cytoplasm divides and forms two different daughter cells. So see, this is the amoeba body and it divides. First the nucleus in the center will divide into two. So the DNA replicate and form two copies. It will divide into two first, and then this will be followed by the division of the cytoplasm, that is cytokinesis. So division of the nucleus and division of the cytoplasm forming a two different individual daughter individual. So daughter amoebae are formed because of the simple binary fission. The second type is seen in the paramecium and planaria, that is transverse binary fission. So transverse binary fission, the plane of the division runs along the transverse section. So cross. Here the macronucleus and micronucleus will be present and both will be first elongated to divide into two, that is karyokinesis, and they form two pairs of macro and micro nuclei, and this will be followed by the division of the cytoplasm. And here you can see this will be initiated by the contractile vacuole. Even contractile vacuole will be dividing into two for the each daughter paramecia. So this we call as the transverse binary fission. So next we have the longitudinal binary fission, which is reported in Vorticella and Euglena. So the organism which has a flagellum will go for the type of longitudinal binary fission. The longitudinal binary fission, nucleus and the cytoplasm divides in the longitudinal axis. You know what is transverse? Transverse means like this cross, and longitudinal means like from top to bottom like this. So the organism that possesses flagellum will go for this longitudinal binary fission. So flagellates like Vorticella, Euglena, the flagellum, one of the daughter cell will be retaining the flagellum, whereas the other one will be producing the flagellum with the help of the basal granule that will be dividing into two during the cell division. So two different daughter individuals are formed in the longitudinal binary fission in the Euglena. Now oblique binary fission. Oblique binary fission, the plane of division will be oblique. It can be at any plane. and it is seen in dinoflagellates and ceratium. The next division is called as multiple fission. Multiple fission, the parent body divides into many similar daughter cells. First, here also the nucleus will be repeatedly dividing. So the nucleus will divide into 2 first, then 2 to 4, 4 to 8, 8 to 16, like that they will be dividing, and many small bits of nucleus will be formed, and this will be surrounded by little amount of cytoplasm, forming many smaller individuals, and this we call as multiple fission. If multiple fission produces 4 or many daughter individuals by equal cell division, the young ones do not separate until the process is complete. They will be together. Once it is over completely, only the individuals will be separating, and this division is called as repeated fission. So they will be dividing 1 to 2, 2 to 4, 4 to 8, like that they will be repeatedly dividing, and so it is repeated fission seen in Vorticella. Then in multiple fission in Plasmodium, how does this happen? Plasmodium is a protozoan. So the malarial parasite Plasmodium vivax and Plasmodium malaria are all multiplying by the multiple fission in our body. So the two stages of the Plasmodium are scission and ootype. So when the multiple fission occurs in the scission stage, the process is called as schizogony, and the daughter individuals are called as merozoites. When the multiple fission occurs in the oocyte, the process is called as sporogony, and the daughter individuals are called as sporocytes. So it is easy to remember. From scission we get the individuals, we call it as merozoite, and if we get the individuals from the oocyte, it is called as sporozoite by the process called sporogony. To understand the multiple fission in Plasmodium, here this is the Plasmodium cell which will form a scission. So inside the scission, the amoeba will divide into different daughter nuclei, many daughter nuclei like this, and they will mature inside the scission itself. Once the maturity is over, the scission wall will rupture to liberate the merozoites. Next, we have the reproduction in amoeba during unfavorable conditions. So during unfavorable condition, amoeba will reproduce in a different way. That is because of the decrease of the increase of the temperature or the scarcity of the food. Amoeba will withdraw all the pseudopodia inside, and it will secrete a 3-layered protective chitinous wall around it, and inside which it will lie inactive. This phenomenon is called as encystment. So when the favorable conditions returns, the encysted amoeba will multiply by multiple fission inside the cyst and produce many minute amoebae, which is called as pseudospodiospore or amoebulae. So the cyst wall will break off, will rupture, and liberating the young pseudospodiospores, and they will feed and they will grow and become different individuals. Next, we have the strobulation. Strobulation is a process by which the adult individual will divide transversely to give rise to a many number of individuals. Most often they will not separate, but after some time they will separate to form different individuals, and this is seen in Aurelia. So this is the Aurelia. The young one will undergo the transposition, and these are the young ones, different Aurelia, and each will become an individual. So that is early Pyra, and this is the both the sides oral view as well as the side view of the Pyra, which is got from by the strobulation of the Aurelia individual. So next is plasmotomy. Plasmotomy is the division of the multinucleated parent into many multinucleated daughter individuals. The plasmotomy occurs in Opalina and Pilomixa, that is giant amoeba. So this is the diagram of plasmotomy. So each individual will be having many nucleus inside. Next, we have the sporulation. So during unfavorable condition, amoeba multiplies by sporulation without encystment. Here the nucleus will break into several smaller fragments, and each fragment will be enveloped by a nuclear membrane and become surrounded by little amount of cytoplasm, and will develop a spore case around it, and these spores will be lying inside the parent body. So once the favorable condition returns, this parent body will disintegrate, liberating the spores, and the spores will hatch out into young amoebae. So next we have the budding. So what is budding? Budding is a process in which the parent body produces one or more buds, and each bud will grow into an young individual. So the buds will separate from the parent body, and they will become an independent individual to lead a normal life. So example for budding, so we have hydra and sponges. So in hydra and sponges, sponges we can see exogenous budding. There are two types of budding. They are endogenous budding and exogenous budding. Exogenous budding is seen in hydro and sponges. So here the buds will be formed from the surface of the parent body. So we call it as exogenous budding. So budding in sponges, the buds will grow from the parent sponge and it will detach itself from the parent body and they will develop into a new sponge. Same way in the budding in hydra. So when the hydra gets plenty of food, the ectodermal cells will increase in size. So you can see the ectodermal cell increasing in size and form a small elevation in the body surface. And this elevation is first it will have the ectodermal cells and slowly the endodermal cells will be pushed from the parent body into the small bud and they form a small bud like structure and at the terminal end they will develop the mouth and this will be encircled by tentacles at the free end, and when these buds are fully grown they will detach itself from the base and finally they will separate from the parent body and live an independent life. These are the examples for the exogenous budding. So next we have the endogenous budding. So in the endogenous budding the buds will be formed within the body of the parent and once the favorable conditions are there, hundreds of buds will be liberated from the parent body. So example for this is Noctiluca. So here the hundreds of buds are formed inside the cytoplasm and will remain within the body of the parent for some time and they will be liberated. So next we have the gemmules. So gemmules formations are seen in the freshwater sponges and also in some marine sponges. A regular and peculiar mode of reproduction in case of the sponges. They form internal buds and these internal buds are called as gemmules. A completely grown gemmule will be seen like a hard ball consisting of internal mass of food-laden archaeocytes. They are called as archaeocytes. During favorable conditions, these gemmules, the sponges will disintegrate to liberate these gemmules. We shall learn fragmentation. What is fragmentation? Fragmentation is nothing but the process by which the parent body will breaks into fragments and each fragments or the pieces will develop into a new individual. There are different types of fragmentation seen. Fragmentation in sea anemone will be different from the fragmentation of the tapeworm. In the fragmentation of sea anemone, here the lobes will be constricted off from the pedal disc and each lobe will grow into a new individual. So these lobes will develop the mesenteries and the tentacles. So they are the internal structures and the tentacles at the terminal end to form the new sea anemone. So what about that fragmentation in Taeniasolium or tapeworm? So in the tapeworm, at the posterior end of the body, we see the mature proglottid or the gravid. So that is the oldest posterior end of the tapeworm body, which is called a strobila. So this will detach itself. They will regularly cut off from the individual body, either singly or in groups, by the process called apolysis. So apolysis is nothing but the separation of the cuticle from the epidermis of the arthropoda. So by detaching themselves, these segments will be transferred into the different host, that is from the primary host to the secondary host through the fecal matter. And here in this inside the proglottis they will have the embryos, and these embryos after reaching the secondary host they will develop into a new individual. So coming to the next topic, that is regeneration. So what is regeneration? Regeneration is nothing but regrowth. So the portion which is lost or the portion which is cut off will be grown. That is called as regeneration. So regeneration is first studied in Hydra by Abraham Tremblay in 1740 itself, and this regeneration itself is classified into morphallaxis and epimorphosis. The first type, morphallaxis, is nothing but when the whole body is cut into smaller fragment, each fragments will be able to develop into a complete individual, whole body will be formed, developed from the small fragment which is cut from the parent individual. So this kind is seen in planaria and hydra. So regeneration in planaria and hydra is of morphallaxis, in which even when the parent individual is cut into several pieces accidentally, each piece can regenerate the lost parts and develop whole individual. So This is morphal axis. The next one is called as epimorphosis. Epimorphosis, here the last parts alone will be replaced. So if this epimorphosis is again of two types. One is reparative and another one is restorative regeneration. In reparative regeneration, only certain damaged tissues can be regenerated, whereas in the restorative regeneration, the severed parts, the cut off body parts, either it is the arm of the starfish can be regenerated, or it is a tail of the lizard, when it is cut off it can be regenerated. That is called as restorative regeneration. Next we shall pass on to sexual reproduction. What is sexual reproduction? So far whatever we have seen under the topic asexual reproduction, now we are going to see about the sexual reproduction. Sexual reproduction involves the fusion of the male and the female gamete to form the zygote, which is diploid. The male and female gametes will be haploid. So two haploid cells fuse to form the diploid zygote, which will develop into a new individual for the next generation. Then this leads to the genetic variation, because two different germinal cells produce the gametes after undergoing meiosis. So genetic variation occurs here. So the type of sexual reproduction seen in animals is called as syngamy. Syngamy or fertilization, that is by the fusion of the two cells, gametes, and also by conjugation. So what is syngamy? So syngamy is nothing but fusion of two haploid gametes to produce a diploid zygote. So depending on the place where the fertilization takes place, it is of two types, external and internal fertilization. External fertilization takes place outside the body of the individual organism, male and the female organisms. The fusion of the male and female gametes take place outside the body of the female organism. Mostly it happens in the water medium. Example, sponges, fishes and amphibians. Internal fertilization, fusion of the male and the female gametes take place within the body of the female organism. Here the higher organisms like reptiles, aves, mammals, internal fertilization takes place. What are the types of syngamy? Syngamy can be classified into the following type: autogamy, exogamy, hologamy, pedogamy, merogamy, isogamy and anisogamy. What is autogamy? Here the male and the female gametes are produced by the same individual, same cell. So same organism will produce both the gametes and they fuse to form the zygote. This is seen in the actinospadium and paramecium. So what is exogamy? So exogamy, male and female gametes are produced by different parents, male parent and female parents, and they fuse. This is called as exogamy. So example, human beings who are dioecious, unisexual animals. Next one is hologamy. Here in the lower organism, sometimes the entire mature organism do not form the gametes, but they themselves behave as the gamete. So fusion of this whole individual, mature individual, is called as hologamy. So this happens in the case of trichonympha. So next one is called as pedogamy. So pedogamy is nothing but the sexual union of the young ones produced immediately. As soon as the young ones are produced from the parent individual, they will fuse to form the next generation. So immediately after the formation of the young ones, by the mitosis process, after coming out of the parent, they will fuse to form the zygote. This is called as pedogamy. Next, what is merogamy? So merogamy, fusion of the small sized but morphologically different gametes take place. We have next isogamy. Isogamy is nothing but the fusion of the morphologically as well as physiologically identical gametes. They are similar, so isogamy. So example for this is monocystis. We have next anisogamy. Fusion of dissimilar gametes is called as anisogamy. So they are morphologically as well as physiologically they will be different, dissimilar. Next we have the conjugation. What is conjugation? Conjugation is nothing but it's a temporary union of two individuals of the same species. During their union, they will exchange a smaller amount of nuclear material called DNA, and then they will get separated. When they are united, they are called as conjugants. Example, paramecium and Vorticella, and some bacteria, prokaryotes are also engaged in this. Next, we have the phases of life cycle. So organisms in their life, they have three phases. They are juvenile phase, reproductive phase and senescence phase. So juvenile phase is as soon as they are born. It is the period between the birth of the individual up to the reproductive maturity. So the period of growth between birth and reproductive maturity is called as juvenile phase. The next phase is called reproductive phase, and this follows the juvenile phase. During reproductive or the maturity phase, the organism will reproduce, their offsprings are produced to reach the maturity period. So on the basis of the time, this can be classified into seasonal breeders and continuous breeders. So certain animals, they are able to reproduce only at certain season, like frogs during the rainy season. They reproduce only during particular season. So example, frog, lizard, most birds, deers, etc. So second type is called as continuous breeders. They will be able to reproduce throughout their sexual maturity. So example, honey, bee, poultry, rabbit, etc. The third phase of the life cycle is senescent phase, which begins at the end of the reproductive phase, in which the degeneration sets. So in the structure as well as the functioning of the body. So this is the third phase of the life cycle. Next we shall proceed to parthenogenesis. Partheno means virgin, genesis means produce. That means development of an individual from the single parent without the fertilization. So development of an egg into a complete individual without fertilization is known as parthenogenesis, and this was first discovered by Charles Bonnet in 1745. So parthenogenesis can be again divided into two types: natural parthenogenesis and artificial parthenogenesis. Natural parthenogenesis happens naturally by itself constantly in nature in their life cycle. So natural parthenogenesis. This parthenogenesis can also be induced for our benefits, and that is called as artificial parthenogenesis. Now let us see the types of natural parthenogenesis. The first one is called as harinotoki. So harinotoki, in this type only males can be produced by parthenogenesis. Example, honeybee. So harinotoki, only males can be produced by parthenogenesis. Teletoki, only females can be produced by parthenogenesis. Example, solenobia. So in amphitoki, third type, both can be produced, both male and female can be produced by the parthenogenesis, and this is called as amphitoki. Example, apis. So natural parthenogenesis may be of two types again. It can be complete or incomplete. So complete parthenogenesis, the individuals can reproduce only by the parthenogenesis only. So there will not be two sex. There is no biparental sexual reproduction. There is no male organism at all. The individuals are only represented by the females. And so complete parthenogenesis alone can take place. They cannot reproduce by sexual method. Next is incomplete parthenogenesis. Incomplete parthenogenesis is found in some animals in which both sexual reproduction as well as the parthenogenesis can occur. So example for this, honeybee. So fertilized eggs will become the queen and the workers, whereas the unfertilized eggs will become the drones. So both are seen in the same type organism. Next we have the phytogenetic parthenogenesis. In the phytogenetic parthenogenesis, the young ones are the larvae. So larvae are the young ones which are emerging out of the parent. This larva at their younger stage itself, they will be able to produce new generation, next generation, before they attain the maturity or the reproductive phase. Then it is called as phytogenetic parthenogenesis. It occurs in the case of sporocyst and radial larva of liver fluke. So liver fluke, the larval stage is called radial larva. This larva itself will be able to produce the next generation by parthenogenesis, which is called as phytogenetic parthenogenesis. It is also seen in the larva of some insects like gall fly. The next we have the artificial parthenogenesis, which here the unfertilized eggs are induced to develop into the complete individual by physical or the chemical stimuli. So we are inducing, so it is called as artificial parthenogenesis. Example, annelids and sea urchins are capable of producing young ones by artificial parthenogenesis. Next, classification of animals based on the development of the embryo. So they are classified as oviparous, viviparous, and ovoviviparous. Oviparous means ovum, ovum, egg. So animals will lay their eggs, and the young ones are hatched out from the eggs. So example for this is reptiles, birds. So reptiles, birds' eggs will be covered by hard calcareous shell. So some of the invertebrates, fishes and amphibians, the eggs are not covered by hard calcareous shells. So oviparous are the animals. They lay their eggs to hatch out their young ones from it. Then viviparous, they will give rise to their young ones. They will give birth to their young ones. So the vivipary is a type of development in which the young ones are born alive after being nourished inside the mother's body in the placenta, through the placenta in the uterus. So majority of the mammals, including human beings, are vivipary. The third type is called ovovivipary. So ovovivipary animals, the embryo develop inside the egg, remains inside the mother's body until they are ready to hatch. That means there is no connection between the mother's body and the embryo through as the placental cord or etc. But they will be getting their food from for the development. The embryo will be getting their food for the development from the egg yolk only. So this method of reproduction, which is similar to the viviparity. Vivipary, that means by laying their young one, by giving birth to their young ones. But the embryos, they will not have any placental connection with the mother, but they receive the nourishment from the egg yolk only. So oviviparity is seen in some fishes like shark.